Welcome to your Week 2 pre-lab. In this lab, you're going to be learning more about Drosophila, the organism that you will use as the basis for Project 1. Shown here is a picture of the laboratory of Thomas Hunt Morgan. Morgan was interested in studying the idea that new species arise because of spontaneous mutations. He decided to use Drosophila to understand if this theory was correct. Shown here is the result of one of his first studies. It might not look like much, but the difference between these red eyes and white eyes really opened the field of genetics. So why did Thomas Hunt Morgan decide to study fruit flies? Well, his reasons were really similar to the reasons we still use today. Fruit flies are easy to grow and care for. In particular, their media is really easy to prepare, which you guys will be doing later in lab. They also breed very quickly. It takes about 2 weeks for them to complete their entire life cycle when kept at room temperature. As a result of the activity shown in the second picture, they also lay very many eggs. Females can lay about 100 eggs per day. Another reason why we use Drosophila is because they surprisingly have a lot of characteristics in common with man. About 70% of the disease-causing genes in human are conserved in fruit flies. Because of that, Drosophila can serve as a model for many human diseases and behaviors. For example, cancer is easily modeled in the fruit fly eye, where genes that control tumor formation cause irregular shapes and folds in the Drosophila eye. There are also Drosophila models for Alzheimer's and Parkinson's, which cause the phenotypes you can see in panel B. Fruit flies can even serve as a model for sexual deprivation and alcohol-related behaviors. As mentioned before, the Drosophila life cycle occurs over the period of several days. There are 4 stages of life in the Drosophila life cycle. The embryo, shown here, the larva, which progresses through first, second, and third larva. Last week you used third instar larva to dissect the salivary glands, the pupa, and then finally the adult. Again, this entire process will take about 10 days from an embryo to develop into an adult fly. In this lab, we will be focusing on the adult flies only. An important skill to learn when using Drosophila is how to sex the flies or how to tell males and females apart. There are a couple easy ways to do this. Females have stripes on their abdomen, shown here, whereas males have a solid bottom. The tip of the abdomen in females is also slightly pointed, which you can see here, whereas males, it's more round. For me, the easiest characteristic to see is that in males, the tip of the abdomen resembles a burnt cigar or the end of a banana, as pointed out here with the red arrows and here in the images. In females, the tip of their abdomen is just one uniform color that comes to a point. Now that you know how to tell flies apart, we can start to get into some more detail. Flies are diploid organisms, just like humans. They have 4 pairs of chromosomes, or 8 total chromosomes. 3 of these pairs are autosomes, and 1 pair is sex chromosomes. Chromosomes 2, 3, and 4 are the autosomes. You can see here that chromosome 4 is quite tiny. Sometimes it's called the dot chromosome. The sex chromosomes are the X chromosome and the Y chromosome, just like in humans. The X chromosome is also called the first chromosome. Females are XX, as shown here, and males are XY. When Thomas Hunt Morgan was mutagenizing flies in order to create new species, initially all of his flies looked exactly the same. However, he noticed one rare fly that looked different than all of the others. What Thomas Hunt Morgan noticed was a different phenotype. A phenotype is an organism's actual observed characteristics. Most of the flies that Morgan observed had red eyes. However, the one rare variant had white eyes. In contrast, a genotype is the set of genes, both normal and mutant, that an organism contains. This specific genotype allows us to observe the phenotype of red eyes, while this specific genotype causes us to observe the phenotype of white eyes. In Drosophila, genes are often named for the mutant phenotype that we have observed. Flies with white eyes have a mutation in a gene that affects eye color. This gene was named white and abbreviated W. Mutations are designated with a minus superscript, and a wild type or normal allele is designated with a plus superscript. Therefore, flies with white eyes are white minus and have a mutant copy of the white gene. Flies with red eyes are W plus and have a wild type copy of the white gene. This can be quite confusing because the genes are named after the mutant phenotype. An easy way to think about it is remembering that all flies have the white gene, but only those with white eyes have a mutation in the white gene. This might take some practice to get used to, so take your time and don't be worried if it trips you up at first. Drosophila genes have been mapped to specific places or loci on each chromosome. Shown on the right are genes on the first, or X, second, and third chromosome. Genes that have been characterized by dominant alleles tend to be capitalized in Drosophila nomenclature. I'll show 3 examples here. Remember, both wild-type and mutant flies each have these genes. However, it's only the mutant flies that show the phenotype that's indicated by the gene name. The bar gene, abbreviated B, is on the X chromosome. The curly gene, abbreviated CY, is on the second chromosome, and the stubble gene, SB, is located on the third chromosome. Based on these names, you can probably figure out what the phenotype of mutants for these genes look like. In Drosophila nomenclature, recessive alleles are generally lowercase. 3 more examples shown here are the singed gene on the X chromosome, the brown gene located on the second chromosome, and the ebony gene located on the third chromosome. Because these genes are recessive, 2 copies are needed in order to see the mutant phenotype that's indicated by the name. Rather than writing out the full name and which chromosome each gene is on, Drosophila geneticists have come up with a shorthand for writing genotype. When writing the genotype of a fly, each chromosome is separated by a semicolon. The X chromosome is listed first, since it's also the first chromosome, then a semicolon, second chromosome, semicolon, third chromosome, semicolon, fourth chromosome. Because flies are diploid organisms, each chromosome can be written as a fraction. The chromosome from one parent is the numerator, and the chromosome from the other parent is the denominator. So, for example, this would be the chromosome that comes from the mother, this would be the chromosome that comes from the father for the first chromosome, and then second, third, and fourth. As an example, a fly with brown eyes, which is on chromosome 2 and abbreviated lowercase bw, meaning that it's a recessive allele, and an ebony body on chromosome 3, abbreviated small e, again recessive allele, would be written as this. The first chromosome is normal. The second chromosome contains 2 copies of mutant brown alleles, which again cause brown eyes instead of the normal red eyes. The third chromosome contains 2 mutant copies of the ebony allele, which causes an ebony body instead of a normal yellow-colored body, and the fourth chromosome is again normal. Completely wild-type chromosomes are often omitted for simplicity. This just makes it easier to write. So you could simply write the genotype of this fly as BW minus over BW minus, followed by E minus over E minus. While you can generally determine if an allele is dominant or recessive based upon how it's capitalized, you can't immediately know which chromosome a gene is on or how it's abbreviated. To do this, you have to use a tool called Flybase. Flybase is easy to get to by simply navigating to flybase.org. And once you get to the main page, you can type in, either at the Jump to Gene or by scrolling down to Everything, the name of the gene you're interested in and simply clicking Search. You can then click on the correct gene, and this will take you to a main page within Flybase. Here you can see the name of the gene, how it's abbreviated, other information, and importantly, down here at genomic maps, you can see which chromosome the gene is on. So now we know that the white gene is on the X or first chromosome. On the right, you can also see a lot of other information that you can access at Flybase, which we'll get into later in the year. For your pre-lab, you are asked to figure out more information about many of these different mutants, including their phenotype, which chromosome they're on, and how the gene is abbreviated. You'll be able to use Flybase to figure out most of this information. If you have any questions or concerns, please don't be afraid to reach out. See you this week!